Hip, hip, hooray for DNA. It provides the key to the plans for making everything in you and me.
Welcome to another past HSC exam question videos. This exam question comes from the Biotechnology chapter. While doing a second, I'll read the actual question. Once I've read the question, you have about 5 seconds to pause the video. Once you pause the video, attempt the question, then press play for me to go over the answer itself. The question, there's 3 parts. A is identify a transgenic species and state its use, and that's worth 2 marks. B is outline one process used to produce a transgenic species, that's worth 2 marks. And C is discuss one ethical issue arising from the use of transgenic species, and that's worth 4 marks. So when you're ready, press pause and attempt the question. Welcome back. Right, so the first part, it's worth 2 marks, and there's identify a transgenic species and state its use. So for the identify, it's just name it, and what I did, and what you probably could have done as well, is name it and then say what combination that is. You know, is it a plant with a bacteria? If so, which plant, which bacteria? And then state its use. So what the actual transgenic species is used for. Why did we do that? What's the purpose of it? So what I wrote is Bt cotton is a transgenic species that has the Bt bacteria gene, so the Bt bacteria gene, inserted into the genome of the cotton plant. Genome is just all of the DNA. So all of the DNA of the cotton plant plus the bacteria gene. So that was the first point. He identified the transgenic species and then talked about the use. So Bt cotton plants are resistant to pests and reduce the loss of crops to pests and increase the profit to farmers and companies. So because they're resistant to pests, that means we have less crops eaten by pests, and therefore farmers make and companies make more profit. So that was state their use, that was their use. So it gets you 2 marks out of 2 so far. For the next one, outline one process used to produce a transgenic species, and that was worth 2 marks. So in this case, it's all to do with, the identify gene, then isolate gene, etc., etc. So I talked about, in my example, I talked about how we can get a bacteria gene, or actually a human gene, into a goat. So what I wrote first is I said the desired gene is identified. So identification was the first stage. It's identified from an organism, and the example would be the antithrombin producing gene from humans. So this is the one we want, and we have it in humans. So we've identified it, and next is to be isolated. So isolated means we actually grab that gene. And how do we do that? We do that by using the restriction enzyme, which cuts out the gene. So now we've identified it and isolated it. And that's the first step. Then what we do is the gene then has a promoter sequence added to it, which allows it to be expressed and cloned to increase its number. So we had one gene and then we increase the number. Actually, we can call it clone, but we can call it something else as well. But we increased the number after and added that promoter sequence. And the last step was to insert that into the actual sheep. So the last step is to insert the gene into the nucleus, supposed to be nucleus of an egg cell. So for example, the sheep egg. So we get that gene and we put it into the nucleus of an egg, a sheep egg. And this is done by directly injecting the gene into the nucleus of an egg. So this is how it's done. So all of this together will get you 2 marks out of 2. Here we have gone through the individual steps: identification, isolation, adding promoter, doubling its number or cloning its number to increase the number of genes we have, and then inserting that gene into the sheep as last step. Now last is discuss one ethical issue arising from the use of transgenic species. So it says one ethical issue. And the ones we raised were stuff like health and medicine, that's 1, health plus medicine. So medical reasons, financial reasons, and social reasons. These were some of the issues, and then we have to debate it, which means we have to discuss, so give the pros and the cons, for and against, for any of the ones that we chose. In this one, I chose the health and medical reasons. So what I did is I said, all came at once. There are many medical and health issues that arise with the use of transgenic species. So here I've identified the ethical issue I will talk about, medical and health issues. Now first, the white will be the ones I did in 4 using transgenic species when it comes to medical and health issues. On the one hand, transgenic species can be used to produce important hormones and chemicals that are beneficial for humans. So by using transgenic species, we can make, for example, insulin using a bacteria. We can also increase the nutritional value of food by altering the nutritional composition of food. So we can increase the nutritional value of food using these transgenic species. So here I've given 2 reasons why we should have transgenic species when it comes to health and medical reasons. Now I give 2 which are against it. So on the other hand, we don't fully understand the health impact of inserting a foreign gene into another organism. The transgenic species might suffer bad health as a result. So here I've just said that the actual transgenic species itself might suffer bad health. So that's a against argument for the health and medical issue reason. So I've said that the transgenic species might suffer bad health, and the next is I also said that it could also cause health issues for humans that consume the transgenic animal or plant. So either the actual transgenic species will be directly affected, or if we eat it, we might have a bad reaction to it, and thereby have our health be detrimentally affected. And it's also worth a mark. So here we get 2 marks for mentioning 2 points when it comes to against, and 2 marks for mentioning 2 points for the mentioned ethical issue, which I chose to be medical and health issues. So where did these come from? They came from these dot points. Outline the process used to produce transgenic species, and include examples of this process and reasons for its use. That was A and B. So B was outline the process, first part, and A was identify transgenic species and state its use, and reason for its use. The other one, so C, came from this dot point. Analyze information from secondary sources to identify examples of the use of transgenic species and use available evidence to debate the ethical issues arising from the development and the use of transgenic species. So here we had these 3 that I discussed in the videos, and then we were asked to just choose one. I chose health and medical. But if you chose the other ones, just make sure you have your pros and your cons. And since there were 4 marks, try to have 2 pros and 2 cons. I hope that was useful.
Thank you for watching.